Do we have this pointer for switching?
I think it's working.
Okay. Okay, good morning, everybody. I'm happy to be here, and thanks a lot to Ima for the invitation to this great place. And also thanks a lot for Justine for the great speech. I think this is definitely state-of-the-art what we have in continuous manufacturing for oral solid dosage forms. So I would like to elaborate a little bit on the way to implement and also to validate a continuous manufacturing process, what we did for direct compression tablets. So what you see here is the installation we have installed in Ulm, which is our biggest manufacturing site at Teva, where we meanwhile manufacture several products commercially also. I would like to a little bit show you around the path. Where did we, or how did we get to this routine manufacturing through process design and also the validation part? So generally, it's important that you be aware, and I think you are that, about what it is behind the process validation principles. So in generally, it starts with the user requirement specification, and then you think about how could your system be designed and everything. And at a certain point, you order the equipment, and then you get delivered it, and then it's installed. And when it is installed, the work starts really for the engineers to do the installation, qualification, and operational. And when it comes to performance qualification, then the process development people and the engineers are linked, because then the product is going to the process. And normally this is not a big approach for the batch process, but if we look for the continuous process, it must be done differently, because for continuous manufacturing you have to consider your product even at the first stages of process design. That's very important to know. So we also ask ourselves the question, why continuous manufacturing? And I'm referring a little bit to other industries. So, and this is just an output of a text which was published on a food journal. And they were also saying that the continuous consistent quality of the product is definitely a benefit for which comes from continuous manufacturing, and also the fact that we have less labor in production. And they mentioned also that it depends on the volume you need to produce. So coming from that perspective, the question is, what is the efficiency we can gain with the continuous manufacturing? So in pharma, okay, of course, we need to consider quality as the biggest aspect, and also the regulatories, but I would like to try to answer this question now to you from our perspective. So the business rationale was for us to have a technology with a high potential of saving in manufacturing costs. That was the rationale behind and the business case justification. We wanted to apply it for a commercial product which was already existing on the market, and therefore we wanted to switch from batch to continuous. No installation in R&D at all. It was set to have an approval requirement with a return on investment of less than 3 years, and after the experience we have, I'm convinced that this is realistic. What is not realistic, what we set as a timeline of 2 years implementation, that was definitely not possible at that time, and I think it's not possible now. General principle for us was keep it simple as possible. That's very important. And what does it mean? So in case of product selection, we decided to go for a product which is really straightforward, which is well established, and it's just, you can see here some points. So the technology should be easy, direct compression. We decided for an NIR installation because we had the NIR competence in-house. That was a huge benefit. The API content, of course, should be high, and the raw material components should not be too many because we try to avoid to have premix and everything. But so it was limited to 5, and that the product quality should be as robust as possible. I think this is self-explaining. Another precondition regarding the business case was to have a high-volume product. And high volume, I'm talking about, let's say, minimum plus 500 million tablets a year. And the raw material characteristics we heard already several times. That is definitely something that needs to be considered also for the dosing. So the strategy we wanted to follow, and we did this, was to stick with a defined batch size, and not only with a fixed batch size. We defined to have a batch size range for that, but at least it was defined batch size. The state of control principle should be applied wherever possible, so we tried to avoid to have the process state in a way that it is too dynamic. So whenever we could have it static, then it should be kept as that. The control strategy was, of course, monitoring all critical process parameters and also the NIR testing on that. Diversion of material was mandatory because otherwise you cannot convince the authorities. That's key. For validation perspective, we defined to go for let's say, a hybrid approach between process-defined implementing into the classical 3-batch validation. In our case, we decided to go not for a real-time release testing, although I would agree to what Justino said, real-time releasing at the final stage brings additional benefit. But for us, it was not part of the strategy. Material traceability was something that needed to be done. So that what was the equipment looked like once we designed it. It's an old picture, but I like it because it's nice, colorful, and you can see everything on it. So you will have 5 components. You see it in the containers there, and we have the transfer pipes, which are red. They're transferring the raw materials to the upper level. From there, they were dosed to the mixer, and from the mixer, they are transferred to the next level beyond where the tablet press is standing. So the general, straightforward approach that could also be applied for other products. So from a process flow perspective, you can see here a little bit more of the details. What I want to show is the elements for process monitoring and control. So we have all the excipients transferred through vacuum transfer, and then it goes to the dosing unit. So each and every component is dispensed automatically. And here we check the dosing accuracy, which is a big hurdle. I will also elaborate later on it a little bit more. From there, it goes now to the right side to the mixing, and there we also check some parameters. But as I mentioned, these parameters are fixed. And we have an NIR where we check the critical quality attributes also. From there, it passes a product valve. Hopefully, normally it passes and it's not linked to the rejected goods, what mostly happens. And from there, we have 2 options. One option is to directly go to the tablet press. The other option is to go to a container, which is later on manually put on the tablet press. So the dosing accuracy, why is the problem? Just in brief, because it's really important to understand, and one of the most critical aspects if you do the full continuous approach. So if you have around about an output of 1 million tablets, and this is the comparison to the bread again, you end up with a dosing accuracy for one of the low dose components with roughly 0.3 grams per 30 seconds. And this needs to be resolved by a balance. And of course, the balance has to be very sensitive to that. And if you compare it to the bread, I think there is not a big issue for the food manufacturing regarding dosing. So this is an additional hurdle that needs to be taken. And why is this a problem? there are two factors that need to be considered. One is the pneumatic issue, because as you work in a closed system and you transfer via vacuum, all the sucking of the material can disturb the balances if you not have a proper de-airing concept. And the other thing is the mechanical decoupling, which is also very important. And we had a hard work to do to really decouple our platform where the dosing units are, because it was always some inexplicable disturbance on that. For example, pallet truck passing by on the corridor or something like that was really giving us a headache, yeah, at the end. So very important. So just on a graph shown how this looks like if you measure the dosing accuracy. You see on the horizontal lines the dosing accuracy, and the bars show the refilling cycles, and just in the middle the big black bar shows the refilling of one of the components, which was a bigger component, so there was 1 kilogram transferred. And whenever this refilling took place for this component, we have a disturbance on all other dosing units for the other components, and that led of course to a rejection of material at the end. Regarding mixing process, we already heard a lot. I think regarding this process, it is very important to know that the residence time distribution has to be known well, and the impact factors on this residence distribution needs to be also developed quite well, because this is at the end assuring the homogeneity of your product. And you yourself need to define in the design phase if you want to vary all those factors, for example, the mixing speed or the output, or if you want to stay fixed. So that's very important. So in our case, we decided to keep it fixed because we did not want to go in the design-space discussions with the authorities. Regarding NIR installation, of course, at the first point, it is important to know where to install. We decided to do it after the mixer, and then we decided for a Bruker Matrix F, which is a quite known equipment, very well working for these applications. We had at the beginning 2 measuring heads, one after the mixer and the second was before the tablet press, but then we decided to remove one after validation. It was enough. So we tested 3 quality attributes, and what was interesting, once we calculated the amount of product we test, we ended up with roughly 7%. So 7% of the product that passed the window that you can see here on the right downside was measured. And if you compare this with the conventional retain sampling testing, what we have in the batch manufacturing process, it's clear that we have a much better insight on product quality and process. But you need to be aware that method development, and I will later on cover this a little bit more, is much more complicated for such a process than for a batch process, and especially the time aspect needs to be considered for this application, because it makes a difference if you measure for the beginning of the batch, and our batch runs also roughly 3 days, or at the end of this batch. So you need to assure that those values are comparable. If you go for the batch release testing, you always have a same sample. Okay, looking at the next stage, which is a little bit more interlinked with validation, it's important to understand that for the batch process, it is mainly required that you have an analytical method validated before you go for the process validation. and then you do the process validation. For the continuous manufacturing process, it's required to have the analytical method validation as part of the process validation, and this needs to be interlinked with the control strategy. That sounds maybe a little bit cryptic, but at the end, you need to assure the measurements and the testing over time are complying your validation requirements compared to single sample testing requirements. And that makes the validation effort much more complicated compared to the batch validation. So if we look at the control strategy, that is also something that needs to be really taken into consideration, because so if you look at this, the text on the right, so there was is a text out of the FDA draft for quality on continuous manufacturing. There is showing that in general, a developing of a control strategy should consider unexpected variations in the process. That means you need to assure that from batch to batch, you have to look at the variabilities that are taking place, and you need to control them from batch to batch. But, and it's also shown here, for the continuous, it's even more critical, because you have to have your disturbances which are taking place during the process under control. And that's then a totally different story you have to consider. And what are the key elements of this control strategy? So we are not anymore talking about process deviations, equipment breakdowns, and maybe some out-of-spec results in IPC testing. We are now talking about input of incoming material. How is this impacting the rest of the process? How do you define your process monitoring and control? And how do you divert material which is not confirming? And that's already mentioned there, that this is some kind of a requirement that needs to be done, because otherwise, whenever you have an variability, you have to divert the whole batch, and that is not making sense at all. The real-time release testing approach is also mentioned, and also what is very important is the setting of specifications, which I will also elaborate later on a little bit more. The impact of equipment, I think this is self-explaining, is having another dimension compared to the process, because for the batch process, you're just looking if it works or not. And if you have some changes, you do a requalification. But what's happened if you have some unexpected disturbances during continuous manufacturing? Yeah, and last but not least, and this is one of the hardest points from our perspective for the continuous, is the integration of the system and the processing of the data you generate, which should not be underestimated, because at the end you're generating a lot of data and you need to evaluate and handle and store this data. This is completely different to what is especially compared to a simple direct compression process. So looking at all those things, you can, in a validation, of course, you go for a process risk analysis. And what is definitely something that has to be in this analysis is to check the suitability for the product, for the continuous line, and also the NIR. Are you able to detect everything you want to see? Or are you just generating some chemometric data that is bringing you not a lot of benefit? And by the way, this is also something that is requested by the authorities if you file the analytical method validation. So the PET hardware is okay, that's clear, and all raw materials impact and on critical material attributes, of course, we discussed and heard in several presentations. The dosing accuracy is key. And also the continuous mixing and homogeneity of the product, of course, is key. In our case, we had to deal with vacuum transfer. That is, I think, not the best, but it's okay if you have a higher API content and it fits to the whole particle engineering, then it's okay. But gravimetric feeding would be, I think, more easier. And the rejection system is something that really needs to be defined very precisely, because for our experience, when we filed, we get questions to how do you assure that the product you're testing with the NIR is completely diverted with the product valve, and that's not an easy answer there, because you have to generate data, and Justine just mentioned and showed it, and I think maybe one of the assessors was participating in one of the presentations, because he exactly posed these questions, and that was not really easy, because if you have the product valve which is close to your mixer, you have the whole data processing from the testing to the valve reaction has to be exactly engineered, and it has to be shown that no product which is tested as a bad or a out of spec product is ending up in your confirming product. That's not easy, but it works, and at the end you benefit with a good process understanding and with a reliable process. So the approach for the quality by design, which we did for the PAT, it's just a principle thing. So of course you start to define which are your critical quality attributes and which process parameters are impacting those, and then you just have to see what technology is the best for that. And when you have decided on that, you can go for the chemometric modeling and the design of this, and also of course the position of the testing and the monitoring principle. And at the end, you should have a kind of control strategy, which is again interlinked with the process itself. So that just shows that it's not just an developing of an analytical method, it's developing of a system. And that's the main difference. So if you just see at this graph here, we are generating data, for example, for API and starch and water content, because we know their impact on the critical quality attributes. Then it's good to have this data, but what are you doing with this data, and how do you handle it, and what are you doing if you have some outliers? Is this already a deviation, or if this has to be handled maybe as a kappa, or is it without any impact? So all these investigations need to be done, and therefore it is very important to set specifications for reacting on those data, and to interlink this with the control system. And here is an example for the control of our dosing units, and it's quite similar for the PAT installation. With the NIR is that we check the dosing accuracy, for example, every second, and when we have a mean value out of this for 30 seconds, and we continuously monitor these values with two limits. One is a warning limit, and the second is an alarm limit. And then you have a statistical evaluation behind that when you are forced to switch the valve to reject the product, and what you can accept if the value is out of warning limit. And that's key of the process design, because you can imagine, and that's what we also experience, if you set those limits too tight, it's normally the easiest way, to be quality-wise on the safe side, just set it to 0.1%, for example, then you will end up with a yield loss in your batch, which is unacceptable high. And then the engineering people are coming and say, okay, why can't we put it to 1%? But if you have 1% variability in your excipients dispensing and you add the other excipients together, then you end up with 3 to 4, and you have immediately a problem with the assay in your blend and in your tablets later on. So that needs to be really critically viewed at and defined. So when it then comes to process validation, we follow the general approach, which I think is everybody doing, based on an FMEA, which is coming out of the process design phase. We implemented, as part of our validation data, we implemented the monitoring data for our critical process parameters, which we have for the whole batch. The blend uniformity data, of course, which are coming from the NIR, was also used, and this was cross-validated with conventional blend uniformity sampling, which means taking samples out of bins. And it was also cross-validated with HPLC testing. That was, by the way, also part of the method validation. So you see the method validation, analytical method validation, and process validation was interlinked during validation batch manufacturing. And it was not, as it is for the batch approach, before process validation to be done. Something different. Of course, we did an enhanced sampling for blend uniformity and continuous content uniformity, because you have a big batch. It's not enough to have only 10 samples for blend. And the operation range and the design space, if you have one, and we try to reduce, as I mentioned, you also have to clearly validate. And one of the ranges we set is a batch size range. So we wanted to be flexible with our batch size, and in our case it was 1,000 to 10,000 kilograms, and this you need to consider in your validation. So we validated 1 ton up to 10 ton with a systematic approach to cover the whole range. But as we heard at the early beginning yesterday, you have to fix your batch size before you start the process. You are not allowed to stop whenever you think it's time for a break or whatever. This would be the easiest, it would be beneficial if you have a breakdown just to say, okay, let's stop the batch and sell that, but it's not possible at all. So you have to fix it before you start. Key. But it's okay, that's not a problem. And we also defined to have a certain range for the output. In this case, it was 100 to 150 kilograms. This was due to technical reasons, because in our case, we had a kind of a big bag for some raw materials, and we needed to interchange the big bags. And to have enough time to change the big bags, we needed to reduce the speed of the line to avoid stopping of this of the process. What is also very important, that is shown here, and I think it's mentioned also already, but I want to again highlight it because it's an impact. The validation of the of start and stop scenarios is something that is new compared to the batch process. So because you have to know what is happening when you start the system, and if you can, it's easy to imagine if you start a continuous process as we have it here, where you dispense the raw materials, you do not start with perfect homogeneity in the mix. That will not work. So it takes some time, and if you can see what you can see here on the graph, it's just simplified the the assay values from starch and the API over time, and in blue you can see the valve action. So we started where we have a high disturbance in the API assay, and then that was okay. And green, you see the starch, it went up and then it went down, and the valve just reacted on that. So what we then did as a consequence, we defined, not only based on this data, but based on much more data, a certain time that is needed until the system is running in a steady state. And again, you need to confirm your diversion of product. That was also said yesterday, and I think it's good to mention it again, because it's another huge approach that should not be underestimated. And clean hold time itself, but dirty hold time is something that is very, very important, because cleaning of such an equipment is a much higher effort than cleaning just the bin and the tablet press. And when it comes to production, the production managers always asking on, let's extend the hold time for the dirty equipment, because we don't want to clean it, we want to reuse it in 4 weeks again. And if you spend 4 days, we are currently in 1 shift or 2 shifts just for cleaning the system, you can imagine that this is an important question. Okay, so intermediate hold time, of course, because you're not testing one, you're not producing one, you're testing 10 batches. Bulk hold time and validation of campaign length is also important, and the most important thing, which is always or very often forgotten, is the revalidation of the models, PAT models. And the responsibility for that, is it still QC, or is the responsibility switching to the manufacturing people, where there is, of course, in most of the cases, no expertise? Yeah. Stage 3, which is the continuous process verification, is for continuous manufacturing a easy stage, because you already continuously check the critical parameters. And I would just show you some pictures of our installation, how it looks and how we operate the system. So it's shown here. On the right side, you see the control units where people can dock to the SAP, to the Bruker, and also to the continuous control unit. This is just a picture of how it looks, and we have installed this in the mixing room as well as in the compression room, because those people in compression also need to know what's happening in the stairs, in the floor above. We operate the system currently still with 1 operator, but we are reducing it to temporary, because it runs very smoothly, and not all the time a person has to be there. So we will reduce to every 3 hours just for changing the raw material bins. Batch size I mentioned. Output is linked to the output of the tablet press, because this is the limiting factor. Otherwise, we could have an output with our system up to 400 kilograms, which we apply for another product. The operator instructions and back documentation is done with electronic batch record. That's also nice, because it can be accessed from everywhere, also from the supervisor room. Raw material, that's base, and the mean residence time, which we calculated for the mix, just from early starting till the end of the mixing, is around about 40 minutes and 130 for the product transferring until it comes out of the tablet press. And that's mainly driven by buffer containers, which are in between. Otherwise, it would be 2 minutes. We have an additional interface for the compression that's mentioned, and another NIR background measurements. We did the big bag containers every 3 hours, and the yield is also an issue. And I have a picture for the yield, because that is at the end supporting the efficiency, because if you have a nice continuous process where you have a reduced quality testing at the end, but you lose 5% of the batch on the way because it's diverting the product too often, then you have gained nothing at the end. So what we have shown here on a graph is that's just a result of the continuous process verification, the improvement of or the reduction of the valve actions, and the resulting improvement of the yield over the past. And we are currently operating with above 99% of yield, even more. So if we look at the benefits, that is not exactly representing what we have as a postulate with the food article. So we see the benefits in manufacturing not in terms of efficiency. We see the benefits in process understanding there, but if you look back now and calculate the manufacturing times, it's mainly a little bit more effort there on the mixing step compared to the batch manufacturing, because the batch manufacturing process in our case was very easy, and now somebody has to do a little bit more than before. But this will see it's still 10%, and if we go to the situation that no operator is permanently at the line, we will also have a benefit there, but that's really the latest state. For compression, it's a little bit better because you do not have the changeovers between all those small batches. And main savings are coming from our calculation out of the QC testing, because there we have a much lesser effort than we had in the past, and it would be even more if we go for the real-time release testing at the end. But of course, you need to invest in this technology, and that takes also time and money. But at the end, we roughly calculate, and this is also confirmed with other products, that the manufacturing costs, and this is, of course, without any material costs, can be improved of roughly 50% if it runs perfect at the end. Okay, just an outlook, and going back to the bakery, they also thought, why to have a full continuous? Maybe it's also worse to have only a semi-continuous. And in their case, they were thinking about kneading process not in a continuous manner, because you have also to, let's say, store the stuff for a certain time for swelling and everything. So they think about having the first step batch-wise and the rest on a continuous. And the benefits out of this are also quite similar from their perspective. And what could this mean for pharmaceutical manufacturing? You can see here, for example, if you're not dispensing all excipients, or you're just having a granule, you could add the granule and the outer phase with the dosing system, and have the NIR installed where you divert then the mix and check if it's okay or not. Go to compression, and then if you look at to the coating, and then if you look for the coating there, if you still have a batch coating process, you will lose at the end all what you gain in the steps before. So you have to separate your continuous big batch into small batches again, and in the worst case, you have to test them all again. And one option there would also be, regarding continuous manufacturing, to have a continuous coating at the end, and this will bring you then the benefits for testing and efficiency on the shop floor too. So just to summarize, process design is key for that, and validation effort is increased. That's absolutely clear. You have to integrate the analytical method validation, and what is should not be underestimated is the shift later on in production from QC to manufacturing. The benefit is the responsibility they have and their awareness on quality they have on the shop floor later on. So in our case it was, and that's also mentioned already, and it's confirmed for us, we have an improved quality, we have a much more robust product. We have reduced manufacturing costs due to QC savings, and we have also reduced cycle time. And from our view, we are now looking on where can we apply semi-continuous approaches to other processes we have in our site and in other sites in the Teva network. Thank you very much.
Thank you very much, Frank. Very clear. Questions? Yes.
Frank, you mentioned that you run the operation with one operator per shift.
Yeah.
I wonder how do you do the verification of the second person? Is that electronically, or is the second person just dropping by to verify?
There is in routine no second person coming to check additional. So the system is validated as it is no second person check needed for that. That was a huge discussion with our quality, obviously, and at the end we could convince them, as we did it also automatically, and there was no process generated that needed to be checked because it was checked by the system and validated in a computer system. We were able to do that. But to be honest, the release process for the QPs or for the quality people has more effort because they have to look at much more data at the end of the batch. Yeah.
Hi. So very glad to see that you have the batch size up to 10K. So I would like to understand what is the process capability, CPK, for the entire process.
So we have the CPK data, of course, for all the critical attributes, and they are far above 2. So the lowest, I think, is 2.0. So it's very robust and constant.
How is that CPK compared to batch, since you're doing batch?
So especially looking at the dissolution part, where we had in the batch some variabilities, it is much better in the continuous process. It was not too bad in the batch process at all, but what we see now is that the variability is much lower. So in the past we had, let's say, CPK from 1.3 to 2 and somebody 3. And now we are constantly on a steady.
Okay. I just had a quick question.
Yeah.
What kind of holdups are you seeing in your system? I mean, how much mass is held up throughout the entire line?
Which one, sorry?
Just wondering about the mass holdup in the line. I see that you got a very fast throughput compared to stuff I'm used to, and you got a long residence time. Do you have a lot of material that's held through the line?
Yeah, so we have at certain points the buffer containers in between to assure that for example, if the compression machine or the tablet press has a stop, that we do not have to stop the whole system, and that's why we need to put some buffer in between after the mixing step and also prior to the compression again. And that was also an outcome out of routine manufacturing, because we knew we had a buffer in the early beginning, about 200 kilo, but we learned very fast that this is too small, because it was very obvious that after a short breakdown of the tablet press, this buffer was full, and then the whole line has to stop. And if I mentioned, if you haven't stopped, you lose material and you have to restart, where you again lose some material, and that's need to be avoided. That's why we increased the buffer in between. Yeah.
Okay, and you run the tallow press extra fast to bring the material and the buffer down, or do you just build up over the course of the batch?
No, the tablet press is running at the level which is exactly the link to the output of the system. So it normally should be equal, but of course if there is some disturbance, then it changes again.
Okay, gotcha. Thank you.
Other questions? Yes.
Good morning. Thank you for the presentation.
Hello.
The question is possible to think to a low dosage API for this kind of process?
Theoretically, it's of course possible, but it's much harder to realize, because one of the limitations, as I mentioned, is the dosing. The second limitation you have is the accuracy of the NIR testing. And this, in combination, gives you a real hard task to build up a control strategy that is reliable, bringing you the product quality at the end without any additional testing. That's from our perspective.
Especially if you are not using gravimetric control, right? Yeah.
Yeah.
Okay. Other questions? Yes.
I think you talked a little bit about it, but just trying to get a little more clarity on pneumatic transfer can have a huge and profound effect on the accuracy of your feeders. What steps did you take to actually mitigate that risk in pneumatic transfer?
What we did is and I think maybe you saw it on the pictures. We did install additional de-airing filters to avoid that the vacuum is directly sucked to the dosing units. So if you install some filters where the air is then sucked through instead of directly going to the dosing units, then it works quite well. But the position of this de-airing installations need to be designed precisely. And to be honest, at the beginning we did not have any of these de-airing filters, and we just added them from time to time on different positions, and but with filters it works. Yeah.
All right, sorry. Is it transfer plug flow, or is it?
It's on a platform.
But sorry, is it plug flow or the pneumatic transfer?
It's a platform, yeah.
All right. Other questions? If not, once again, great presentation, Frank. Thank you so much.
You're welcome.
All right.